All right, welcome to Chapter 2 on mitosis and meiosis. So, some of the key concepts that you should understand by the end of this chapter are listed in this first slide. So you should be able to differentiate between a genome, DNA molecule, chromosome, homologous chromosome, and sister chromatid. So hopefully by the end of these chapters, these words will make sense. If they don't, that means you need to study this material a bit more, because these concepts will be tested on the exam. Understand the difference between ploidy, haploid, diploid, triploid, and number of chromosomes. So if you have 10 chromosomes, 20 chromosomes, and then how to actually calculate both of these numbers from a karyotype. I want you to be able to describe what occurs during the various stages of the cell cycle, which is not the same as mitosis and meiosis. And then also understand the stages of mitosis and meiosis. So what are the stages, and then be able to sketch the behavior of chromosomes at each of these points. Understand why meiosis but not mitosis generates genetic variation. So the daughter cells of meiosis have different complements of DNA than the daughter cells of mitosis. So why is that? Relate how the structure and activity of chromosomes determines the inheritance of genetic information, and then finally describe how defects in meiosis lead to issues in public health. So if things go wrong in meiosis, what are some diseases, syndromes, etc., that occur because of this? So DNA carries genetic instructions for the development, function, growth, and reproduction for all known organisms and many viruses. So I'm assuming you know all this information from your intro bio class, and this is pretty straightforward. So why do humans become human? Why do chimpanzees become chimpanzees? Why do dogs become dogs? They all start from a single cell, and the reason is because their DNA complement is different. So the dog genome contains instructions for creating a dog. The human genome contains instructions for creating a human. And so what is DNA? A DNA molecule. So typically your cell contains a large number of DNA molecules, maybe not a large number, but on the order of 50 to 100, depending on when you look. Depends on the organism, but the first thing to know is that DNA is organized as DNA molecules. So that's an important thing to remember. So what is a DNA molecule? It is two polynucleotide strands that coil around each other to form a double helix. So the double helix, the fact that DNA is a double helix, is something that you should also know from intro classes. And so there are two strands, and these two strands, of course, are complementary to each other. So if there's an A on one strand, there's a complementary nucleotide on the other strand. If there is a C on one strand, there's a complementary nucleotide on the other. So each of these strands encodes redundant information, because if you know the sequence of one strand, you can specify the sequence of the other strand. And so there is a length of this DNA molecule. So how many base pairs? So a base pair would be the combination of these complementary nucleotides. And so if you counted how many of these are, that is the length of the DNA molecule. So this is 2 base pairs, 3 base pairs, etc., etc. And so a typical DNA molecule is actually quite long in the cell. So it's, on the order of 100 megabases. And so there's the length of this DNA molecule, is on the order of 1 meter. So if you stretched out this DNA molecule, normally it's in a cell, which is microns in length, but it's actually quite condensed. And so it's important to remember, and it's important to really wrap your brain around the fact that this is a physical object within the cell. And so the DNA molecule is an important subunit to start to think about. And so the subunit of DNA contains nucleotides. So there's an A, a C, a G, and a T. And of course, these are complementary to each other. So the C binds with the G, the A goes with the T. And then we can simply write out one of these strands in order, so on and so on and so forth. So another key background is that there are 2 main types of cells. So there's prokaryotic cells and eukaryotic cells. So prokaryotic cells include small things like bacteria and archaea, and these are primarily haploid. So each bacteria typically only has 1 copy of each chromosome. DNA is organized into a single circular chromosome. There's some exceptions, but typically, if you take the entire genome of an E. coli bacteria, it's all condensed into one single DNA molecule. And instead of this DNA molecule being linear, it ends up binding back on itself to form a circular molecule. Eukaryotic cells, so protists, plants, fungi, animals, we of course are eukaryotic. These are primary diploid. So diploid means we have 2 copies of each chromosome, and then DNA is organized into multiple linear chromosomes. And so instead of there being a single circular chromosome, you have a large number of linear chromosomes, and they're different sizes. And so this chapter is mainly going to be covering what happens in the eukaryotic cells. So the transmission of genetic material, and that involves either mitosis or meiosis. So the meiosis is the process leads to the production of gametes. These are sperm cells or egg cells, depending on your sex. And this is what is used for reproduction. Where mitosis leads to the production of 2 identical cells. So this is important for development. So when you are developing from a single fertilized egg into an entire organism, this occurs in the womb during development and occurs while you're growing, you're actually going from 1 cell to trillions of cells. And so the way that happens is that cells divide over and over and over again, and they use this process of mitosis. So this chapter, again, is just going to cover these 2 essential processes, meiosis and mitosis, in a lot of detail. So the chapter 2 content, so this will be organized into 3 main parts. So first is the basics of DNA organization in the cell. Second is what happens during mitosis and meiosis. And then part 3 is the health consequences caused by defects in meiosis.